Well, first, I would like to thank Doug and everyone at Santa Cruz Works for giving us the opportunity to talk about RealSeq biosciences. So we're going to change directions a little bit from the two previous presentations. So we're an RNA detection company. And our idea is that we want to take our basic research work into point-of-need testing. So my name is Sergio Barberán Soler. I was born in Mexico City, and I came to the US to UC Santa Cruz to study a master's and then a PhD in molecular biology. And then I went to Barcelona to do a postdoc, also working on RNA and detecting RNA. And then that brought me back to Santa Cruz, where I work at a company called Somagenics, a small biotech company here in Santa Cruz, also detecting RNA. So in all, I have around 20 years of experience with detecting RNA. And in 2019, me together with Ann Schultz, which is the COO of RealSeq Biosciences, Brian Johnston and Sergei Kazakov, we decided to found RealSeq Biosciences. So the idea is I bring the RNA experience into the company, and Ann brings the business experience. She has close to 20 years of experience in life sciences and also in venture capital experience. So what is RealSeq? As I was saying, we focus on detecting RNA molecules. The way we funded the company is that we got seed money from the NIH. We got 2 grants from the NIH to develop technologies for the life sciences to detect RNA. That seed money from NIH allow us then to have innovative products and services to detect RNA molecules for life science research. And the key thing here is that part of my idea with science is that we should make it as simple as possible. So we want to have simple and accurate technologies. But the second goal of the company is that we want to take all of those RNA expertise and bring it back to diagnostics. So we want to have simple and rapid diagnostics that use RNA. And at the beginning, we want to focus in neglected diseases, orphan diseases, these kind of diseases that are endemic to some regions that no big pharma has focused before. We have been in business for 2 years. Last year, we already had $500,000 of revenue from our commercial services and products. And the problem that we're trying to tackle here is, as all of you know, unfortunately, COVID has shown that diagnostic testing in the US and in the world is not up to the game. We have been in this pandemic for almost 2 years, and testing is still a big problem, takes a long time. There's not sufficient testing, and there are many problems. And you can imagine, if that's for COVID that has changed everyone's life, you can imagine that for endemic diseases, like for example, there's valley fever, that is a fungal disease that just happens in a region in the Central Valley and in some places in Arizona. Testing is still worst. There are no tests that they can use. It's really difficult for the clinics to diagnose that disease. But we believe that RNA should be the key for that diagnosis. The problem right now is that because no one has invested time and expertise in developing that, RNA diagnostics are high complexity tests. Things need to go to a highly qualified personnel with really expensive equipment. So that's the problem we want to tackle. We want to use RNA diagnostics for easy, fast diagnosis for orphan diseases. So our solution to that is what we call ribomarkers. So ribomarkers is just, we detect really small pieces of RNA that allow us to detect diseases, infectious diseases, much faster and much more accurate and sensitive than other technologies. What we're doing right now is a collaboration with the Valley Fever Institute at Kern Medical, and we're developing a test that allow us to diagnose valley fever, this fungal disease that I was talking about. So what we do is we take a serum sample for a patient, and we can help them to determine if they have a localized infection or a disseminated infection. And the RNA advantage is really clear against the DNA. So we want to use these ribomarkers for valley fever first, but then we want to develop a platform that can move into other technologies. So the next steps for the company will be, we really need to move into a Series A after having seed money from NIH. We're ready for the next step. We need to expand our team. We're fortunate enough that in Santa Cruz, UC Santa Cruz, the RNA experience is really vast. So there's a lot of people that we can hire. We want to be CLIA certified to be able to test for many diseases here in the Santa Cruz community. And we want to get our first diagnostic test to the market. And this will allow us to develop a platform that then we can expand into other diseases, neglected diseases, or whatever pandemic comes next. So we hope that in a couple of years, we're able to have 2 products for valley fever. As I was saying, we want to be able to diagnose if a patient has a localized or a disseminated disease, but we also think that RNA and testing in general needs to be point of need tests that you can do at home, that you can do on the field, that you can test anyone out there. And with that, that's my presentation. I would just like to thank everyone. Thanks, Doc, again.
Well, thank you. And, you know, the valley fever is something that's really close to us because it impacts agricultural workers, right?
Correct.
So we love our agricultural workers here. The other thing is that people know, a lot of people know that at UCSC they completed the Human Genome Project, and they know that, you know, the COVID virus was sequenced there as well. What I don't think a lot of people in Santa Cruz know is that there's this guy named Harry Noller who was kind of the guy at UCSC and the world for RNA research. And it's everything that we're doing, the mRNA, everything that's been used to treat COVID is because of some of the work that Harry Noller has done. Right? I mean, I'm sure you've probably mentioned.
You're correct. I mean, UCSC, the RNA group at UCSC is world known. Harry Noller is the best one, but there are many researchers out there. It's one of the best places in the world to do RNA research.
Sure is. Well, again, thank you. And we really appreciate the work you're doing. Keep our ag workers safe and find those other sort of rare diseases and viruses out there. And let's treat them as quickly as we can. Thank you so much.